Okay, hello and welcome to everyone who has joined us online. I'm Mila Pollock, Executive Director of Libraries and Archives at Cold Spring Harbor Laboratory and the CSHL Center for Humanities and History of Modern Biology, which organized today's event. The laboratory is a nonprofit research institution located on the North Shore of Long Island, New York. It dates back to 1890 when it was founded as a marine biology station. Research at the lab has led to 8 Nobel Prizes and significant discoveries in genetics, molecular biology, and medicine. The institution's research contributed to our understanding of cancer, ALS, depressions, and autism, among other diseases, and it produced the first effective treatment for the devastating genetic disease, spinal muscular atrophy. Cold Spring Harbor Laboratory is now playing an important part in research effort to help understanding treat COVID-19. We are delighted to gather virtually to bring these two accomplished scientists and authors into conversation. Professor Siddhartha Mukherjee is a cancer physician and researcher at Columbia University. His 2010 book, The Emperor of Maladies, A Biography of Cancer, was awarded the Pulitzer-prize for nonfiction. Today, we will be discussing his more recent book, The Gene, An Intimate History, which won international awards and was recognized by the Washington Post and the New York Times as one of the most influential books in 2016. The Gene was the inspiration for the new Ken Burns documentary of the same name. In conversation with Sid today is Matthew Cobb, professor in the School of Biological Sciences at the University of Manchester. In addition to his research on genetics and physiology of the sense and smell, Matthew has published extensively on the history of genetics, including his 2015 book, Life's Greatest Secret, and his latest book, The Idea of the Brain, The Past and Future of Neuroscience and Smell. He was one of the screen experts in Gene. My colleague, Richard Sever, is a moderator for this event. Richard is Assistant Director of the CSH Press, an Executive Director for the journals CSH Perspective and CSH Protocols. Richard is the co-founder of the critical resource known as bioRxiv and medRxiv that have allowed thousands of scientists around the world to make their COVID research available as quickly as possible, and obviously with free access. We are delighted to welcome Sid and Matthew for a conversation. You can submit your questions through the QA box at the bottom of the Zoom screen. And then Richard will present the questions to Matthew and Sid after a conversation. I'm so grateful to you, Sid, and to you, Matthew, for joining us today. Sid and Matthew, please.
Thank you very much, Mila. Sid, good to see you. So we haven't met in the flesh, but we've had a few chats over Zoom in the last week preparing this meeting. I wonder if we could start at the beginning with the title of your book. I read somewhere that there are perhaps 20 definitions of a gene, depending on the scientist who's using the phrase, depending on what they're interested in. So I wonder if you could tell us, well, what you think a gene is and what it does, and why there's this difficulty that people have in coming up with a definition, which might appear to be quite a simple question.
Well, it's my pleasure to join you, Matthew, in this conversation, and thank you for Cold Spring Harbor for hosting this. It is actually quite clear that many people have different definitions of the word gene. People have tried to define this word, understand this word. I wanted to use, in some ways, I wanted to use in the book every definition or cover every definition. A gene is a unit of heredity. A gene specifies a trait. A gene is usually the code that carries the code or the material, the instructions to make a piece of RNA, which by itself can be functional, but more often gives rise to a protein which carries out a function. A gene carries information from parents to offspring. A gene is a piece of a DNA. Originally, it was thought to be a continuous piece of DNA. We know that that is no longer true, but it is a piece of DNA. A gene in a virus can be carried, as we know from COVID-19, from SARS-CoV-2, a gene can be carried in RNA. So it's really a piece of nucleic acid, which carries the instructions to build an RNA, which in turn carries the instructions to build a protein. So it is like looking at the same thing through multiple perspectives, multiple directions. No definition is complete. No definition is absolute, but they're all correct. And so I felt that we needed to embrace all of them, and different aspects of the book and the film emphasize or go into the idea. And it's a historically evolving idea. The analogy that I like to make is, the word atom means different things to different people. An atom is a unit of matter. It is the constituent element of a molecule. It is the indivisible piece of matter. But of course, we know that the atom itself is made of divisible pieces. And so much like the atom, the gene, the definition of the gene is evolving. It will continue to evolve. But the fundamental piece of it, which is that it is a unit of heredity, and that it is carried across generations in the form of a nucleic acid, remains basically unchanged.
I think we can leave the rest of it, the niceties, to the philosophers of science. Maybe
that's right.
They've got plenty of time to logic shop. I was watching the comments on Twitter as people watched in particular the first episode of The Gene, but both episodes.
Which you're in.
Sorry?
Which you are in.
Which I'm in, but that's not what I was watching. And a number of people that I saw on my timeline were very surprised to learn about the way that, in particular in America, the history of genetics and the history of eugenics, that is, of improving the human stock, as they would have said at the time, was so intertwined. And I thought that was a very good part of the film and of your book, because your book deals with that as well. I wonder how surprised you were. I mean, can you remember when you first realized this? Were you taught it at university or at school back in India? What was when did you find out about that, and what did you think?
Well, that goes back to the process of writing the book, and we haven't. What's interesting is that I haven't really talked much about, aside from this forum, talked much about the process of writing the book. It was both surprising and also a kind of hidden aspect of American history. The standard way that eugenics is taught is as if it grew out of nothing, like a ghost in Nazi Germany, and in fact grew as a part or an organic part of Nazism, and grew late perhaps in Nazism. But when you study the history and when you look at the archives, you realize that that's completely not true. that in fact the eugenics movement has three distinct phases. The first phase, first of all, maybe even four distinct phases, I would say. The first phase goes back all the way to Plato and Aristotle, and people who for a long time wondered whether heredity could be manipulated. But of course, they had no idea of genetics. They had no idea of the gene. They didn't know the word gene. They didn't know the concept. So far from it. And Plato writes about it very briefly. He thinks about an ideal race. He thinks about it mathematically. He thinks about mathematical ways of breeding people so that they would get the best possible outcomes. So that's the ancient history of eugenics. And then it mostly vanishes from at least from philosophical writing. But then there are these three contemporary phases, and we only know about the last of them because it's been popularly taught. The first phase was in the UK, in England, or in Great Britain, I would say, where as geneticists began to understand the relationship between genetics and selection, people like Francis Galton and others, very luminary characters, began to wonder whether they should select for the best possible humans using what they would now call positive eugenics. In other words, breed the best and make a kind of a nation of elite. There is there was a famous saying that that somehow or the other, by breeding this national elite, they would work themselves out of what was becoming very clear in Great Britain and its colonies, which is that somehow or the other there was a fear of racial degeneration, and that they would work themselves out of racial degeneration by breeding a national elite using genetic techniques. So that's where the seed comes up. Francis Galton, of course, an eminent statistician, an eminent a geneticist, I would say, and a biometrician, someone who loved to measure things, the shape of your skull, the size of your head, how far your eyes were apart.
He had a particularly creepy habit of wandering around the United Kingdom with a piece of paper in his pocket and a pin, and he would measure. He would note down how attractive he thought the women were.
That's right. That itself might make him not particularly attractive to these women, but that would be that was Francis Galton. Galton was a measurer. He was a statistician. He was a mathematician. He loved measurement, and he was also a cousin of Darwin's. And so somehow or the other, he thought that he could take what Darwin had understood about selection and what English farmers were doing with selecting animals, sheep, cows, pigs, and somehow translate that into human beings. Then, so that's the birth of positive eugenics, and it must be said that there were people in England, notable scholars, among them G.K. Chesterton, who really warned against this idea. This was not while the Galtonians and the English elite embraced the idea. This was considered very mainstream. It was like considered taking a walk in the park, and considered part of what one should do to make a better nation. And so while these people were moving along, there were critics. There were critics like Chesterton and others who said, well, wait a second, there's something wrong, morally wrong with selecting humans. So that's the first phase. The second phase moves us to the United States. In the United States, eugenics takes a very, I would call, a Yankee practicalism. The practicalism is, let's forget for a second about all the philosophical conundrums. Let's just do it. And so in the United States, it was done. And by done, I mean, there's a famous story which opens the book, which is the story of Carrie Buck, a woman who was basically, and her mother, they were basically sent to a confinement colony in Virginia so that they would not reproduce and produce more children. And eventually Carrie Buck was sterilized, medically sterilized, so that she would not produce any children because it was thought that she was a so-called moron, a definition that was made based on very, very quirky and largely unsubstantiated rumors. And this was a big movement. This is not a small side note. People like Ellis Havelock and others wrote extensively in the United States that this was the way to move the country forward to take away the racial dilution that was happening because of the immigration to the United States. So that's the second phase. To me, that was the most surprising phase. I had heard of Bolton, but I had not really heard of the Carrie Buck case. The Carrie Buck case was that Carrie Buck herself appealed to the courts, and the case climbed to the Supreme Court. And there was a chilling opinion issued by none else than Dr. Holmes on the court, which said 3 generations of imbeciles is enough, thereby enabling the state to sterilize, to remove the ovaries of Carrie Buck, so that she could not have any more children, and therefore breed further imbeciles. This was a surprising history. And then finally, this kind of malignant phase of eugenics moves to Nazi Germany. Hitler himself was fascinated by the idea. He was fascinated by the idea even as a young man. And then, of course, when he ascended to power, he and his various associates would begin to practice not just sterilization or positive eugenics, but the ultimate perverse form of eugenics, which is to kill people, exterminate people based on what was thought to be their racial and genetic flaws. And so it was particularly the second phase, this American phase, the incubation of eugenics in America, that surprised me.
So do you think that's something that we need to teach at school and certainly at university, that people need to understand these roots of their science?
I think it's very important to teach that, because I think there was a normalization, and this process of normalization enabled the ultimate incarnation of eugenics in Nazi Germany. The normalization occurred by or was propelled by scientists and intellectuals and doctors and writers ranging from, you know, Alexander Bell and H.G. Wells and people who you would not imagine were associated.
And everybody was eugenicist in the early 20s
exactly.
and 30s.
And there were few critics who said, wait a second, this is, you know, Oliver Wendell Holmes. There were only few critics, as I said, Chesterton among them, who said that there was something morally incorrect or something morally smelly about using human beings as objects or subjects in this kind of vile experimentation. So the normalization of eugenics as a consequence of genetics is something I think we should teach everyone, particularly because we're, as you know, and we'll talk about it a little bit more, we are coming to an age in which we are once again able to read and write human genomes in a way we couldn't before. So the technological brink has brought us to a point where we could reinvent, to some extent, a new form of eugenics, and it would recapitulate perhaps some of these very vile ideas about how to make human beings better. And so I think it's something we should really.
Even before we get on to the most exciting modern technology of CRISPR, which we'll discuss shortly, there was, at the moment, we're actually carrying out a form of soft eugenics. We're changing the genes in the human gene pool by genetic counseling, in that people are given information. It's then up to them as to whether they want to have a baby, but people can then decide. They can make a choice, and we are effectively influencing the spread of genes in the population by providing people with information where they can then decide, do they want to have a child or not? And that relates to what I wanted to talk about next. So what you talk about the Buck case, and what struck me in reading your book, and it's what you're renowned for, is the way that you weave in personal stories, including stories from your own family, with the history. And the bit in the TV series, in the two programs, that particularly I found the most powerful was the scene where the young English woman was going to get the result of her test for Huntington's disease, which, if it went one way, if she had the mutation, then effectively it was going to be a death sentence, and if it went the other way, then she was free. And that was an incredibly tense and powerful sequence. I wonder which was which part of the programs, which aren't an exact copy of the emotional bits and the personal bits of your book, but which parts of the TV programs you found particularly telling and you were pleased to see on screen?
So just all those points are important. I mean, the television series that I did with Ken Burns is not a facsimile or a copy of the book.
No.
It's very different from the book. It's I would say we had a long conversation about it, and it's really a complement to the book. They work as yin and yang. In the television series, we decided that people, that the long history of genetics is covered in the book, and we would concentrate more on real stories, on contemporary stories of genetics. And that was a very complicated decision, because Ken Burns, of course, is also a historian. But we felt that, if we went into the depths of the science, we would miss what genetics what's happening in genetics today. Now, that said, what no one knows, I think, and this is the first time I'm talking about this, initially, we had the idea of me going back to Calcutta or Kolkata and meeting again my cousin, who has a very severe variant of schizophrenia and is permanently hospitalized. And we decided against it for several reasons, but that was one idea that was in the film, and that would have required an enormous effort. My biggest concern about that was consent. I couldn't imagine how he would give consent and how he would understand consent. So we decided that, with people with mental illness, the idea of consent is very important, and being on a very public forum like television would have been extraordinarily complicated. So even if we could move a television crew, and I agreed to do it, I was very hesitant about the idea of consent. And so that's certainly something we can talk about. Now, the question you asked was, what were the moments for me that were incredibly moving? I will say both. On one hand, finding, as you mentioned, the woman with Huntington's disease, finding out her diagnosis, and but equally, the child being treated for spinal muscular atrophy. One child in that family dies. And then they have a new child. They have another child. And devastatingly, they find out that this second child also has the disease. And you can imagine what's going through that family's dynamics or its brain. One child has just died, and the new child has been born, and he too carries the mutation. And in the middle of all of this, of course, the drug for spinal muscular atrophy, which is really a genetic drug, comes around, and this child begins to walk.
Miracle.
It really is. It's really one of these examples. of miracle medicine. When I saw the footage of that child walking, I really nearly broke down and cried, because it was just incredible. And finally, I would say that, spinal muscular atrophy is a relatively rare disease. There's beautiful and very important footage of a young African-American man with sickle cell disease, very difficult to treat normally, because the only things that we really have are oxygen and pain control. Being given a bone marrow transplant with a modified gene that will essentially relieve the symptoms and enable him to not have sickle cells anymore. And we follow his life. And in one of the last scenes in the film, he's sort of playing Uno with his family, and his requirements for going to the hospital and being transfused, and the pain that accompanies it have diminished dramatically. So all of these success stories were really gratifying.
Yeah, I think that the case of sickle cell anemia, or sickle cell disease rather, as it's called these days, is so important, because there are so many people who are affected by it, generally, but not entirely, African Americans in America, or Afro-Caribbeans in Europe, and of course the massive African population. And yet often it's completely kind of overlooked in favor of other diseases that can be treated. But as you say, with the possibility of the new gene editing techniques, then with the right care, this is a disease that can now be cured in principle. And we saw an example of that. I wonder if we could say, move on therefore, thinking about precisely CRISPR and other gene editing techniques. In general, for the reasons you've outlined, because of the eugenic past of our science, people are extremely wary. And there is at the moment a general agreement that we should not be editing the germline, the eggs and sperm. So editing a cell that's going to produce your blood cells, I think in general people are okay with that if it's safe. They don't think there's an ethical problem. But the ethics of changing the future generations is seen as being that's the wrong thing to do. And the opprobrium that fell on Dr. He when he carried out his experiment on those, turns out to be 3 little girl female embryos 18 months ago, demonstrates that. However, what possible harm could it do, say, to remove the part of the Huntington gene that causes Huntington's disease? What if we could do that accurately and safely? Where could it go wrong? Because we know what the gene does in me and you, and we'd simply be turning it into that version. Do you have a view on this? It's a complicated, difficult question, but we're all going to have to face it.
Yeah, we're facing it now. There are several comments to make. Well, first of all, just an anecdotal comment, again, just to give you a flavor of how the film was made. When the film was actually complete and edited, when Dr. He announced the birth of what he calls Lulu and Nana, the twins that whose embryos he had edited. And I called up Ken and I said, this has got to be the beginning of the film. So we reopened the fully cut film and inserted back in this episode because we couldn't ignore it.
No.
So that's just a little piece of I would an anecdotal piece. But look, there are the one very clear line to be drawn here is that the genes that if we are to start editing human genes in sperm and eggs or sperm and egg producing cells, the first thing is it has to be safe. Yeah. Like all of medicine, the first concern is safety for the child. And remember, this is an unborn child. The fetus cannot speak. The parents are speaking on behalf of the fetus. So it's an unborn child. So the first concern is safety. And again, around that concern is the understanding of consent. Again, the fetus cannot consent. The embryo, Lulu and Nana, did not consent to anything. And in fact, we now know that the consent forms for the consent for that procedure was very badly done. The second idea I think that's important is that the world has clearly understood that there has to be some sense of extraordinary suffering associated with that genetic disorder, whatever it might be. And it really has to be extraordinary. It has to cross that threshold. Now, what's extraordinary to you may be different from what's extraordinary to me. So there are gray zones in here. But in general, the idea that there would be that I think that most people would agree that Huntington's disease in its fulminant form is a disease of extraordinary suffering for patients. So that's the second question, or second strong line in the sand that we've drawn. The third is, so as I said, the first is safety. The second is extraordinary suffering. The third is that we should have an enormous degree of confidence that altering the gene will indeed alter the physiology or the future fate, genetic fate, physiological fate of the individual. And we don't know this for many genes, actually. So you may think we know this for many genes.
Yeah.
And there's a popular press idea that you know it for many genes, but you don't. And this is an extraordinarily important point. We do know that women who have a higher risk of breast cancer may have a mutation in the gene BRCA1. What we don't know is how many women with BRCA1 develop breast cancer in the future. So we have very strong, we now know actually for BRCA1 in a much better way because we've done these studies. But for most genes, we don't know. We usually look at genes retrospectively. If you have the disease, do you have the gene? But we rarely have looked at genetics prospectively. If you have the gene, do you have the disease? Yeah. So this point that I'm making, this third point I'm making, is a far from trivial point, because genes are modified by the environment, they're modified by other genes, they're modified by chance, they're modified by various other things. So you really have to be confident that the genetic change that if you do make for a disease of extraordinary suffering, that you're making this in good faith, that you will relieve the disease. And finally, I would say that you need to make sure that there isn't another alternative. So for the child with sickle cell anemia, if a bone marrow transplant with minimal conditioning, I'm a hematologist, this is what I do, with minimal conditioning, so that it does not set that child up for a risk of future cancer, for instance, that you can do an autologous bone marrow transplant and cure sickle cell anemia, then why go into the germline where there may be safety concerns? So I think that these are some of the criteria that we are developing over time. And as I said, safety first, extraordinary suffering, the strong conviction that correcting the gene in the embryo or sperm and eggs will actually correct the disease. And finally, that there is no other alternative, and there is no other, that we're not doing this in a forcible way. I think keeps us at least 1 degree away from the specter of eugenics, but it's not the solution. We still will need to keep monitoring and continuously trying to understand why and how and where we're going with this technology.
So to ask you almost a kind of political question, how can we decide that? Because it's too important to be left to the scientists or the physicians. It's this is a problem that the whole of humanity is going to have to face. How can we ensure that there's sufficient involvement of the population, that people realize the opportunities and the dangers, and can help as a community, as a global community, come to some kind of agreement as to what is acceptable?
Well, I think the first step is education, understanding what technologies are available and what the safety of those technologies are and how we've used them before, what the track record is and for those technologies. So that's the first step. And that's why I hope the book and the film are really able to bring those two things to the front. So that's the first step. The first step is, as I said, a technological education. The second step, I think, is to create a consensus. And it's very, very important because we have some models for this. When recombinant DNA was first discovered, actually by my former mentor, Paul Berg, among other people, there was a furor, there was a very great worry that we would start playing with genetics in a way that was not safe. And so many, many meetings were organized. Among them, the most famous of them is the Silomar meeting, which really put some strong boundaries around what was permissible and what was not permissible. What's important about this Silomar meeting, I think, is that it was not just a meeting of scientists. Journalists were invited, legal scholars were invited, bioethical debates were encountered, the general public was invited. So this is what we're talking about, is the human genome. It is the thing that you and I possess in common. And it is not up to lab scientists or doctors in white coats to decide whether to move forward or not. So I think there is going to be, as I said, a lot of education and a lot of consensus building before we should do these experiments. And the question really arises if we should do these experiments. One could make a strong argument that there are some diseases where you could use genetic counseling and what is now called prenatal genetic diagnosis to not have a child with, for instance, carrying the Huntington's gene or the BRCA1 gene. So should we go that way and let this specter of genetic alteration on the side, or should we allow for diseases of extraordinary suffering the possibility of editing the human genome in sperm and eggs?
So shortly we're going to be taking the questions, because I can see them piling up. Richard will be assembling them. I wonder if we could, before we move on to that, if we could shift gear. I'm interested in the way that you write. I mean, I've written a book which partly overlaps or contains a bit of what you wrote. And it was very interesting to see the differences between how we approach the topics. And your way of writing, it seemed to me, as I've already said, had a much more personal, almost artistic touch. And I wondered whether, I mean, you're a physician, you're a scientist, but you're also a physician. And physicians, as I think you've just eloquently described, have a richer view, I think, often of problems than somebody who is simply, like I do, studying flies. You've got to, seriously. And so I wondered whether you thought, I'm interested in the link between the way medicine, science, and art. So on a personal level, your wife's an artist. She's a famous artist, Sarah Z. Have you ever worked together? Do you think
we work together extensively privately, in the sense that she reads everything I write. I'm sitting actually in the studio, in Sarah's studio in the house, where I'm looking at, you can't see them, massive 8-foot by 8-foot paintings that she's just finished. But we work together privately. We critique privately, but we very, very rarely have done public events together. We probably have done 2 or 3, partly because we just don't think that, the public events end up being, have ended up being too far of a crossover. But and it's very hard to cross that in half an hour or 45 minutes. If we did a public event and it was 2 hours long and we'd bore people to death, we could certainly do it. But we've done very few. We've done a couple. So as far as medicine is concerned, I think of myself primarily in writing, and not as a scientist writer, but as a physician writer. And that's very different. It's why, for instance, the book may, to some people, move fast or even gloss over some aspects of genetics, but really focuses quickly on human genetics. And I wanted this to be a book about human genetics as much as it is a book about the history of genetics, and therefore the title, it's an intimate history. An intimate, by intimate I mean not just the history of my family, but the history of our human family. And that was actually a common critique of the book, that it was this book should have been called a book of human genetics, which I'm fine with, and it's correct, in fact. But there is, when you write a book, you have to choose. Otherwise, you would write 2,000 pages, and this book was originally about twice its length. And so a lot of things were moved into footnotes. A lot of things were moved into side notes, et cetera, et cetera. It's like making the film. Again, you have to choose.
It's like making a work of art in a way. It's like,
exactly.
my experience of writing, especially the editing, is it's like sculpting. What a sculptor says is, you're removing the things in the block of stone, and you've got to remove the stuff from it. And a sense, sometimes you feel, yes, I've just got that right. That's what it's supposed to be like.
Yeah, and also, the stories of genetic scientists is obviously interesting and important. But as you've done yourself, I've read your book, of course, and loved it. But a fly geneticist would write a very different book than a hematologic geneticist. I mainly study the genetics of blood disorders and try to cure them. I do transplantations, among other things. And so it's a very different history. It's a very different story. And I think that understanding those differences is crucial to reading a book. A book written by a physicist turned geneticist. What is Life? The famous Schrödinger book is very different from a fly geneticist writing about genetics, a human epidemiologist writing about genetics, and in this case, a doctor writing about genetics and writing about his own family history.
Okay, Richard, have you been going through the questions? Do you want to fire some of the matters?
Inevitably, there's quite a few that have been coming in. It's great that you've actually touched on some of them. There's a lot of queries about ethics. I suppose the first most important question in some respects is, will this all be on YouTube? To which the answer is yes, it will. But I guess there's a lot of follow-on from the queries about ethics that you've been asking, Sid. One of these was, do you think we need a change in teaching? The way we teach genetics, the way we teach biology 101, this notion that it comes as a surprise to learn that there was a prehistory to eugenics before the Nazis, and the extent to which geneticists themselves were entwined in the broader population. So do you think we really need to think again about how we teach genetics and biology when people first come across these concepts?
It's an important question, Richard. And I think that, not to plug this book, but to plug a whole series of books, there should be a curriculum in which basically every college student in a science track, and perhaps even in humanities, is taught something about genetics, because Darwin and Mendel and the Great Synthesis and the subsequent enormous strides and leaps that follow are part of human history and human culture. They are not just part of scientific history and scientific culture. So I know that the gene is now part of the curriculum. So while you're making Punnett squares to figure out how a certain set of genetic traits might be transmitted, you can also read about the social history and the prehistory of genetics, and they complement each other. You understand that this Punnett square you've made on the blackboard or this Mendel, when you learn about Mendel's experiments and what he did statistically, began to have enormous relevance in human social history, in human legal history as well. And I generally encourage, when lots of people have reached out to me, educators, professors, and I say, you're not going to use this book as a textbook, but use this book as part of your curriculum to emphasize to people who understand genetics, to understand that there's something beyond the textbook, and that the things that you're learning in the textbook have consequences outside it. Matthew, how do you teach a fly genetics course, and what do you do about?
Well, we have history of science courses as well that teach this, and in the history of when we're teaching genetics, we probably don't go into it as much detail as we do, but my colleagues at University College London have done exactly this. Steve Jones, the famous geneticist, popularizer of science, he's now an emeritus professor, but he still has, and he's done this for about 20 years, he teaches their students the eugenic past of Francis Galton, and they have to do this because Galton is linked with UCL. They have Galton lecture theatres, and they've had a huge row about what they should do about all this legacy of a man who, as you said, is extremely important because of his contribution to statistics, but there's other things we can learn from him, bad things we need to learn, mistakes we mustn't make. So I certainly at UCL, that's been at the heart of all of the genetics teaching for about 20 years, including the teaching they do for the medical students. So a part of the UK scientific tradition has learnt this for many years.
I should say one more thing, which is that when we started the film, actually, we were going to start the film with Victor McKusick. Victor McKusick is very well known as the person who started one of the first human genetics programs in the United States. And he would often go by himself to Amish County to study human genetics. And he was among the first people to talk about genetic disorders, again, a tradition that began in Great Britain and then was moved to the United States. So I've been encouraging people to take a much more sort of global view, around a sculpture in the round view of genetics. Richard, maybe we should go on to the next.
Yeah, so there's a couple of related ones here. One is, do you think that things like your book will encourage more nuance in the understanding of nature versus nurture, which is obviously being, that debate has been going on for a long, long time. And most geneticists will say it's a bit of both. And there are reasons to be optimistic that people think that message getting across, but then there's also reasons to be pessimistic that people seem to be reverting to more genetic drivers of certain tendencies, which we'd like to talk less about.
Well, the book I tried to be fair to this question of nature versus nurture in the following manner. I think that the debate has become polarized, and in becoming polarized has lost its bearings to some extent. If you have the mutant version or the disease-causing version of the Huntington's disease gene, then it is very, very likely, nurture aside, that you will develop the disease in your adulthood. That is a genetically dominated disease. In contrast, there are other things that have, where the environment modifies genetics so dramatically that you may have a very mild version of a disease, even if you carry the genetic predisposition to it. And in fact, breast cancer is one example of this. We now know that many people, that there are modifier genes, modifying behaviors. We know this about common things like heart attacks. We know that coronary disease is caused, there's a strong genetic component to it. It is caused by multiple probably hundreds of genetic variants accumulating, as it were, in a person's genome. And yet there are people walking around, rare syndromes, in which just one gene will give you a risk of 50 times the risk of the population of coronary disease. So I think this is an arena where a nuanced discussion about what exactly nature means and what exactly nurture means. And it's not always going to be nature plus nurture. Sometimes it's going to be strongly dominated by nature. Sometimes it's going to be strongly dominated by nurture. And many times, or more often, it's going to be some spectrum in between. And so there has to be, I would say, some specificity to the conversation. When you say nature versus nurture, the question I usually ask is, nature versus nurture for what? For what gene? For what condition? Are we talking about breast cancer? Are we talking about coronary disease? Are we talking about Huntington's disease? Are we talking about spinal muscular atrophy? Are we talking about, a variety of other diseases where genes are extraordinarily strong determinants of whether the disease happened, and no matter how much you try, the genes will drive the disease. I think it's important also at a human level, because if we begin to confuse the idea of nature versus nurture, then we also ascribe blame to people who cannot modify nurture to defeat nature, and they feel defeated by it. There are conditions still where the genetics are so dominant that no matter what you do, you're not going to reverse it. You're not going to suddenly be able to change. And it takes the blame away from, for instance, parents. A parent of a child with a severe cystic fibrosis can certainly protect that child from infections, but the child will and may manifest the various complications of having two mutations in the two alleles of the cystic fibrosis gene. It's not the fault of the parent, it's not the fault of the child, it is the deck of cards, the genetic cards that child was dealt. And so I think having a nuanced conversation, whenever people say, is it nature or nurture? I always ask back, well, regarding what? What is the gene? What is the condition? What is the ailment? What is the trait that we're talking about? And in some cases it will be one, and in some cases it will be other, and in many cases it will be in between.
Yeah, I mean, so that's one of the points you made there was that it depends on which disease you're talking about. Another question that's come up in as being is how much of it is a problem that the things that we're looking at in sort of population genomics are informed by the fact that most of the studies of genomics have been done on Caucasians, and how problematic is that when actually most people in the world aren't, and the diseases that they may be affected by are different.
Well, so I think it's a big problem. I think that we have several diseases where there are strong genetic components, and therefore we need to figure out, we know that there are strong genetic components because they are passed on in a hereditary manner. And it would be very, very helpful to have a much broader genetic database in which we would understand this. So I'll give you one example. Prostate cancer among African Americans is one example. There are several others, beta thalassemia and others, where our genomic databases are quite poor in capturing populations which are not Caucasian. And so those are extraordinarily important. I think we are finally seeing projects out of, for instance, China, West Africa, India, and other places. And often there's an overlap between what's in the Caucasian population, but the overlap is not 100%. And so there are new genes or new risk alleles that are found in non-Caucasian populations, which are important to understand and their biology, and also understand what kind of risk they confer. So this project is broadening, although it's taken longer than one would have hoped.
Right.
Yeah,
So another related question, sorry, Matthew, another related question to that, which I'd be interested in both your perspectives on, is the fact that now lots of people are having their genome sequenced. This is becoming much, much easier. So Matthew, you talked about genetic counselors, but the possibility for almost everybody to have their genome sequenced at a trivial cost, and the privacy implications for that. There's been a couple of questions around the issue of society and politics, and how does that change things, and whether one can, given how easy it is to sequence somebody's genome now, really expect any form of privacy going forward, and do we have to think about new legislation to protect people?
Well, so the privacy concerns are becoming progressively important, because I should say that we've entered a new phase of genetics, and I'll let Matthew comment on this in 1 second, but we've entered a new phase in genetics in which I would say for human genetics, the first genes that we began to link to disease were genes that have a powerful contribution to the disease. So single genes that have powerful contributions to disease, cystic fibrosis being one example, breast cancer, BRCA1, and others being other examples. So these are genes, I call them shove genes or shove effect genes. These genes shove you towards a particular disease or fate or a particular disease. What we're finding out as computational power grows is that there are many human diseases which we knew were hereditary, in the sense that they were carried in families, where the effect of any single gene is quite minor, but the accretion or the risk of multiple genetic variants adds up to give you the full risk of the disease. A great example of this is coronary artery disease, again, heart disease, very common. There are people who have, in coronary artery disease, about 4% of patients with coronary artery disease will have identifiable mutations in single genes that will have an enormous effect and increase their risk by a very large percentage. Most people with coronary disease it's a result of genes and environment, for instance, diet, exercise, hypertension, smoking. But genes do play a role in this disease, and we now know from a whole bunch of studies that, in fact, many done in the UK, that the genetic component cannot be underestimated. That there is a genetic component, but that genetic component is carried by multiple genes, not one, hundreds, perhaps even thousands of genes, each giving you a little bit of risk. So I call these nudge effects or nudge genes, each giving you a little bit of a heightened risk. Now, the problem is that as we enter this new phase of genetics, where we are beginning to break apart the egg of the nudge genes, privacy becomes extraordinarily important, because you can get, if you sequence your genome, Richard or Matthew or me or Mila, if we were to sequence our genome, we would begin to find risks and quantifiable risks for diseases like coronary disease, for conditions such as obesity, for diseases such as breast cancer, and many others. And therefore, the legal, if an insurance company were to find this out, you can imagine that they could do things that were not, that we think would not be allowed under current understanding of insurance. So the privacy question has become very important, and the possibility of genetic information leak has become very important now. Matthew, any thoughts about that?
Well, yeah, this is, as you say, the UK is particularly strong in this area. We've got something called Biobank, where there's about 100,000 people across the country, including my sister, actually, who have volunteered to have all of their physiology and their medical records linked to their genomes. And that way, researchers are able to look for correlations between very small variants, as you've just indicated, these nudge variants in their DNA and particular conditions. And at that very high level, you're going to end up getting insight into the kind of processes that are taking place. We don't have the worry about medical insurance because we have a socialized medical system where it's free to the user, which is great. But I think for those people who are the viewers and listeners who have carried out one of these tests that proves they're 20% Viking or whatever nonsense it is, just remember, nothing's free. And you've paid for it. And in fact, you've given them your DNA, and you've also given them a lot of details about yourself, and that's their business model. These databases from the companies that are sequencing, not a full sequence of your DNA, but they're looking at bits of it, are going to be used in various ways. So who controls the databases becomes incredibly important. And in the UK, there was recently a huge row because this biobank database was going to be exploited by Google in ways that weren't entirely clear to many members of the general public. So I think
again, remember, this is your DNA. I mean, this is the most personal. I mean, it's more personal to you than your credit card. It would be like distributing your credit card information to a company and saying, take it and use it as you will. Just give me back, I don't know, whatever, it might be a small compensation for it.
Yeah. But of course, you're leaving it everywhere, as we know from every episode of CSI. You're throwing this stuff about. You shake hands. Well, we don't shake hands at the moment, and so clearly there's all sorts of fantasies about, well, why don't we just have everybody's DNA sequence when they're born? I mean, that could help the medics and it could help the police. And if you've done nothing wrong, then you've got nothing to hide. I mean, I don't think there's many people who seriously are advocating that, but I think that's an aspect, the sociological, political problems associated with our genes, with our intimate life, as you rightly say. We've all got to watch that like hawks, I think.
Absolutely, yeah. So, moving on slightly from Matthew's utopian view of the future, there's been a few questions to both Sid and Matthew about more personal ones about your own approach, your own approaches to information and writing. So one of them was, how do you both keep up with the deluge of information, and how does this inform your writing process and your choices about narrative in terms of, the cases you choose? I mean, you mentioned some of this earlier, but the examples you choose. And so there's a few questions about your writing process. I wonder if Matthew, do you want to speak to that?
Well, let's Sid first. He's the Pulitzer-prize winner.
My general rule is that I try to emphasize, my general rule is that no 2 pages of a book should pass without encountering a real human being. That's just my own rule. I use it, whether that's me, whether that's a scientist, whether that's a patient, whether that's a discoverer, whether that's a piece of social history. That's my general rule of writing. Because I believe that, these things have human consequences. And as I said, every book about every subject is different, and you just have to accept that. There will be, during the editing process, there are people who go away from the book, people who leave the book, because you can't have, if you write about every single person, the book becomes a kind of what I call name salad. Name salad is a very, very bad thing to do to a book, because you then become unreadable. It's an unreadable book. You produce a document that no one will read, and therefore has really no consequence in contributing to the conversation. So you have to make choices and be ready to be critiqued by people about those choices. People are disappointed in those choices. But you have, as a public figure, as a writer, as someone who writes, regularly for the New Yorker, for the Times, not a day will pass by when I would get 50 letters by email, or 40 or 30, saying, you know, what a wonderful book, thank you so much. And then 10 of theirs will say, why didn't you include my mother? You know, what about my great uncle who, you know, did this? And I'm generally humble about it. I generally thank them gracefully, and I say, look, I mean, you know, that book couldn't be written. That book would be 2,000 pages. There's an encyclopedia. And also, the other philosophy that I have is that, and I often say, you know, I often tell people this directly. I say, if you're the kind of person who opens my book and looks for your own name in the index, this book is not for you.
We've all done that, Sid.
Exactly. So if that's the motivation, this book is not for you, because you know more about the topic than if you're in the index, you know more about the topic than I do. So that's my general set. I'm being facetious about it, but you can't write that book. That book, it cannot be written. It will be an encyclopedia which is unreadable. And so you make choices, and you have to sit with those choices. I mean, you know, I'll give you one example of this very particular example. I happen to know Marshall Nirenberg's family very well, and they're collaborators. His second wife's son is Jonathan Weissman, a very, very great scientist. He's actually a very close friend of mine, for reasons that have to do with the structure of the book, you know, of course, Nirenberg, of course, was a person who deciphered the triplet genetic code. But for reasons that have to do with the structure of the book, you know, of course he was mentioned, but the entire story of the triplet deciphering was condensed into one paragraph. When I look back, I thought to myself, gosh, you know, perhaps that was a wrong decision, but you have to live with those decisions.
Well, they can use Matthew's book for that then, right?
Exactly.
Thank you, Richard. I was going to say that. I think that the key point is what kind of Sid just indicated, that you end up with kind of 2,000 pages or a huge, great big thing, and then you've got to edit it. And the secret of writing is in rewriting, because the first draft or even the second draft of virtually anything is crap, and it's full of all sorts of baggy bits, and it's only by going over it and over again and chipping away like the sculptor and removing the bits that you thought were important, but then you realize that it makes the reader trip up, or it takes them down a side track where it doesn't go anywhere. And you've got to keep the story going, keep the pace going. And you know, sometimes we've all got little factoids, little stories, little darlings that we love, and we don't kill them all, but that rule, kill your darlings, I think is a very good one for, you know, the factoid that you have chiseled out, that you've discovered and is really exciting. After a couple of months, you look at it and you think, nobody actually cares except me. Let's get rid of it. It doesn't enlighten us.
I'll give you another example. I mean, it was actually an example that relates to Cold Spring Harbor. And it's an instructive example. You know, Barbara McClintock was so far ahead of her time as a scientist that it was impossible to fit her into this book. She invented a whole lineage of genetics, which we are now finally discovering, particularly around transposons and CRISPR and so forth, genetic modification. And I tried and I tried and I tried. I tried to fit her, jam her in here, jam her in there. She was just so far ahead of everyone else in terms of thinking about genes that it was really impossible to fit her in. So the best I could do is put her in a footnote. Now, if you're a Barbara McClintock fan, and I am personally, you would say, how dare you put, you know, of all people, Barbara McClintock in a footnote. But there was no way, because she was so far ahead of things. I mean, she could have come back in a kind of CRISPR episode, I suppose, but there's just, sometimes you just have to be, you know, you're stuck with the, I mean, no one knows this, of course, because the Barbara McClintock fans will now start writing to me tomorrow and say, you know, why did you, you know, here's a perfectly good place that you could have put Barbara's work, or Dr. McClintock's work, I would say. But she was, again, so far ahead of her time in thinking about mobile genetic elements and genetic recombination, blah, 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 that I tried and I tried. There was a big paragraph on her. I finally moved it to the footnotes. There was nothing else to do. And I have to now live with it. Maybe in another edition of the book, which will come out, now that we know more about CRISPR, we'll have a chapter on mobile elements, and she can come back to life in this book.
So we're just about coming up on time, but one more question for you both before I hand you back to Mila. A couple of people have asked this, which is, who are the figures that inspired you, and what are the things that, I'm presuming you're working on, but what are the things that you would recommend that other people read? I mean, I'm right behind your right shoulder, I see Cicero said. So I don't know if that's on the list, but I think people are very interested in the figures that have. I know
there's I mean, Matthew and I were just talking about it. There's apparently a whole meme going around of the books that are in the back of people when they're on Zoom chats. So I'm going to look back and see what among this is of interest quickly. I thought this book was interesting, actually. This is Kat Arney's book called Herding Hemingway's Cats, How Our Genes Work. It has a lot of stuff on epigenetics, which is interesting and is coming to life.
Well, that's great, because that was a question that we didn't get to. So somebody can read a book on it instead.
Yes, you can read a book on that instead, yes.
Matthew, any thoughts?
Your books are less visible.
Well, yeah, that's just piling up. There's all sorts of. This is the UK edition, but it's Transcendence, which is available in the US as well, by Gaia Vince, and it's about humans' evolution through fire, language, beauty, and time. So it's all about human ideas and our human evolution in a way, in a fully social way. Fantastic book.
Right. Well, it's interesting to know that you're reading books rather than just wading through documents at Cold Spring Harbor. But thank you very much for your answer to those questions. And I'm sorry to all the people who asked questions who we didn't have time for. And I'll just hand you back to Mila.
Richard, I want to add one more question because it's sort of related to me, and I'm very happy that, from participants, we received the question, and the question was to see it, and they said that many original documents are shown in the documentaries, and how can viewers find these documents so we can look at them? And if they're publicly available. It's actually to both of you.
Well, so Mila, this is to thank you as well, because the Cold Spring Harbor archives are a very valuable resource. In fact, one thing that is a very, very valuable resource is that you can actually search. You don't need to. I'm sure you'd welcome them to go to the Cold Spring Harbor, but a lot of those documents are online and they're searchable. And in fact, they're searchable. They're actually very well indexed. So for instance, you can, if you'd like to, there are a series of interviews with various geneticists and biologists. There are also original letters, which are housed there. You can read, if you wanted to read Rosalind Franklin's notes, you can access them, not directly through the CSHL, but through a portal that the CSHL, the Cold Spring Harbor Laboratories, has. You can see her first impressions of photograph 51 online, never leaving your home. And Matthew, I know you've worked directly at the archives, so tell us a little bit about that.
Yeah, well, I was working there on the life and work of Sidney Brenner, great British geneticist who won the Nobel Prize for his work on C. elegans. And just going through... I met him
several times, actually.
Sorry?
I met Sidney, Sir Sidney Brenner, several times.
Very funny man.
Yeah, he's a very funny man.
Yeah. And I worked at the archives. I was looking at his correspondence, in particular with Francis Crick, and the sometimes rather risque postcards they'd send each other in the 1960s. And all these you can see them online, but after the pandemic is over, then go and visit, if you can get a chance to visit the archive, to actually see the original document is extraordinary. To feel it, to see what has been said. I mean, you know, amazing documents that are present there. Absolutely fantastic resource.
Oh, thank you to both of you. It's actually for me like a pleasure to hear this, because we have so many scholars coming and use it, and we're open to the public. So everyone who wants to come to see it, they can see it. For those of us who have enjoyed so much seeing your books and watching the documentary, it has been fascinating to hear these personal reflections. Also, Matthew and Sid, thank you very, very much to sharing with us actually your secrets of writing and to answering to all participants' questions. Thank you, Richard, to work so hard on this. And thank you very much to the participants. But I want to emphasize that it is recording and it will be available, your conversation and all event on YouTube, as well at our cshl.edu website and on the website of the Center for Humanities. Thank you very, very much for participation.
Thank you.
Thank you.
you.
Bye-bye.
Bye-bye.